This podcast is for informational and educational purposes only and is not to be considered medical advice for any particular patient. Clinicians must rely on their own informed clinical judgments when making recommendations for their patients. Patients in need of medical advice should consult their personal health care provider. From UPMC Children's Hospital of Pittsburgh, welcome to That's Pediatrics. I'm one of your hosts, John Williams, Chief of the Division of Pediatric Infectious Diseases and Director of the Institute for Infection, Inflammation, and Immunity in Children, or I4Kids.
And I'm Steph Dewar, Vice Chair of Clinical Affairs in the Department of Pediatrics, Co-Director of the Pediatric Residency Training Program, and a member of the Pediatric Hospital Medicine Division. We're happy to welcome Dr. Kemaladewi, who has extensive training in human genetics and development of genetic therapy with a focus on neuromuscular disorders. She's interested in understanding the molecular mechanisms underlying layers of pathophysiology involved in muscular dystrophies, including muscular atrophy, regeneration, fibrosis, and nerve damages. She has a robust portfolio in the development and evaluation of therapeutic genetics and uses a technique known as CRISPR to correct mutations and modulate disease modifier genes in mouse models. Dr. Kemaladewi, welcome to That's Pediatrics.
Thank you for having me.
Well, we're excited to have you here, and I was embarrassed to find out that I did not know you previously. And so I'd just love for you to share with us a little bit about what you're involved with here at Children's at your lab.
Yeah, so I came to Pittsburgh about 1 year ago. I started in February 2019, and my lab pretty much used genetics as a tool to do two things. One is to study neuromuscular phenotypes, and second is to develop therapeutic interventions for rare diseases. And the first disease focus for our lab is LAMA2 deficient congenital muscular dystrophy, or LAMA2 CMD in short. And this disorder is caused by mutations in a gene called LAMA2 that is important for muscle integrity and nerve myelination. So we use a technology called CRISPR activation to develop a mutation-independent approach, which is the key here, mutation-independent. And how do we do that? So we use this technology to activate a gene called LAMA1, which is structurally similar to LAMA2, which is the mutated gene. And by doing so, we should be able to compensate for the lack of that mutated gene. And my lab are testing this in a mouse model in a sort of like thorough, dedicated preclinical studies to evaluate the efficacy and safety. And on top of that, we are also testing this approach in patient-derived cells to see if we can eventually bridge this from mouse to closer to clinical application.
So this is really interesting, Dewey, that you can basically, if I understand you correctly, you're activating a different gene. You're actually not fixing the gene that's defective. You're activating a related gene to sort of take up its function.
Correct.
Okay, that's really interesting. So a question about this type of muscular dystrophy. There are a number of different kinds of muscular dystrophy. You know, how common is this type? And do we know the genetic bases of the other types? Could you take a similar approach for these other kinds of muscular dystrophy?
So I started this journey just like many other researchers in the field when CRISPR became popular or famous in 2012-ish. So we wanted to use it to correct the genetic defects in LAMA2, the underlying genetic defects as a therapeutic approach. Later, we realized that mutations found in patients are actually very heterogeneous. In a sense, you can track 200 patients who have 200 different mutations throughout the gene. So that fact alone, together with the rarity nature of the disease, makes us think that if we want to target this disease, we should develop an approach that can be beneficial for all patients, irrespective of their mutations. And that's where we came up with CRISPR activation and LAMA1 as a target. In terms of other muscular dystrophy, for sure, so the most common muscular dystrophy that affects pediatric is Duchenne muscular dystrophy, which is caused by mutation in dystrophin gene. And there is a similar gene called utrophin that is expressed during embryonic stage. And similarly, so LAMA1 and LAMA2 is kind of similar as well. And we all know about SMN1, SMN2 for SMA. So yeah, so I'm not sure how common it is if you're talking about like the whole genetic disease, but it's actually a good numbers if you think about it.
So it sounds to me like the value of your research is focused on this particular disorder, but also could be translated to approach other diseases in a similar way.
Yeah. Yes. So I know that other researchers are trying to use similar approach to target haploinsufficiency cause conditions. Basically, yeah.
So I don't know anything about CRISPR. I don't know what it stands for. I don't know how it works. I don't know how long it's been around. So maybe you could help me understand that.
The abbreviation, I'll probably have to Google it. It's something with a repeat and stuff like that, but I will come back to that. But CRISPR pretty much is it's a stretch of viral genome that are captured and found in bacteria. So when virus attacks bacteria, when bacteriophages attacks bacteria, this bacteria has Cas proteins that will digest this foreign genome that is from the virus. And part of them were captured and integrated into the bacterial genome. So it's part of the bacterial and archaeal immune system, basically. And scientists then tweak this system and make it a powerful tool that can help answer many biological questions, including development of therapies.
So
it has been around for a while, and I think when I started my postdoc in 2012, that's when the field just took off. And I think part of it is because some scientists actually try to make it a system where many researchers around the world can use in a simplified manner.
Thank you.
So how would then, if you could get this to work in the human cells, then how would it work that you would put those back into a human?
Yeah, so
in other words, what's the translation from cells in a dish that have been repaired essentially to now be functional to then putting those back into the human?
Yeah, so there are multiple ways I can tackle this question. I can say it in a way that what kind of CRISPR trials are out there right now, right? So in 2019, people combined CAR-T with CRISPR. So you can actually modify T cells and introduce that. And I think the latest news that I've read is that it proves safe in a clinical trial in cancer patients. That's one way to do that, ex vivo. And another trial that is out there regarding CRISPR in patients is conducted in Cambridge and Dublin on genetic blindness, liver congenital amaurosis, and the results are eagerly awaited. So the difference being targeting specific tissues like eyes versus muscles is, of course, muscles is a very large organ. So the challenge with targeting diseases like muscular dystrophy is, of course, the delivery. So in my lab, we use a adeno-associated viral vector serotype 9 or AAV9 that targets muscles and nerve to deliver the CRISPR components to mouse and hopefully at some point humans. And the AAV9 itself is also being used in a lot of clinical trials. So we are sort of like just taking what people learn and combine this together.
So are we at that point here yet in Pittsburgh, or are we still working towards the use with patients?
We're not at that point yet. This is always a difficult question to answer. So what my lab is going to, or is actually, I've been planning to do is to treat the mouse model in a long-term manner. And then along the way, we are going to test to see the efficacy of this approach. So we're doing a lot of assessments in the muscles and nerves, as well as how it affects respiration of these mice. And then towards the end, we will look at what kind of immune responses that these mice develop to Cas9 proteins, because the Cas9 proteins is a foreign protein, so we have to take that into account as well. And also, if we are targeting just muscle and nerves, but if we are injecting the virus systemically, maybe this approach have detrimental effects in other organs, and we don't know yet. And that is one thing that my lab is going to tackle in the next few years.
Well, it's interesting that I think maybe it's a feature of the modern era that we both expect things to get into humans very quickly, and that we, oftentimes they do, but you're describing, a prolonged period of going from where you are and to getting it into humans and getting it working. But, Tom Starzl, the father of liver transplantation right here in Pittsburgh, did many, I think dozens or scores of failed liver transplants before it started working, partly because they didn't really understand the immunology and a lot of complicated things then. So, I guess we shouldn't be in too much of a hurry to make you start treating humans tomorrow.
Correct. And one thing that that I also want to highlight is that CRISPR, in the end of the day, is a tool, right? So, yes, I want to develop it as a future potential therapeutics, but I think it also has a lot of potentials to understand the disease itself. And another aspect of my lab that I'm actually very interested in, very excited because it's a new thing, is to tackle another aspect of the disease that which is the peripheral neuropathy. So the LAMA2 gene, in addition to being expressed and important in muscle, it's also important for Schwann cells myelination. However, not all patients develop peripheral neuropathy, but some do. And it is very prominent in the mouse models. But this is kind of like an underexplored aspect of the disease. So my lab is trying to exploit CRISPR activation to convert patient-derived fibroblasts that we know what kind of genetic mutations they have to induced Schwann cells and model myelination defects on a dish. So that's another new area that we would like to explore.
So I'm wondering, we have this sense of urgency to get treatments to patients, but I'm concerned that this COVID pandemic has interrupted your work.
Yeah. So as I mentioned earlier, so my lab we just celebrated our 1-year anniversary when COVID-19 hit. And I think as a new PI, when we started our lab, we sort of like optimized everything, recruit people, and try to get assays optimized. And I think my lab was at a sweet spot where we lay all the groundworks necessary for future research. And then we were almost at that point when we're trying to test valuable samples before COVID-19 hit. But it doesn't matter as long as everybody is healthy. So all my lab members are healthy and in a good spirit, and they are still very passionate about their project. And I think we can, whenever the lab opens again, we can get like not a
well, I certainly appreciate that perspective. I agree with you. It is very challenging times for all of us, but you're right. The key is that we're alive and well, and those that we love and care for are also alive and well. So it's a great perspective for all of us.
I agree. It really highlights to, your experience of just what you're saying, the balance that we collectively are trying to strike between an urgent pandemic, a virus that is circulating and making people sick and causing deaths right now, versus a disease that you researched that's a very serious, life-affecting disease, and at the moment, nothing's happening, right? So we sort of have to, as a society, as scientists, and our leadership at Children's here, at UPMC Children's, both the clinical and the research leadership are working very hard on this, at finding that balance between keeping people safe from COVID-19, but allowing other research to sort of continue and resume in clinical care so that progress can be made in other areas.
Yeah, in such a way, like in a really weird sense, and I told my lab as well about this, that pandemic like COVID-19 and rare diseases share a few things in common. They happen infrequently, but when that happens, it affects global community and lead to significant economic loss, right? So we should be in the same mentality in tackling this too. If that sounds overly optimistic, sorry.
No, I agree. That's a very good perspective. So you mentioned that you came here in February of 2019. Can you share with us a little bit about your path and your journey to this type of science and subsequently to Pittsburgh?
This type of science, I've always been in genetics department, or now it's called division, but I've always been surrounded by geneticists, and that gets into my mentality, I think. And since my PhD, I've always been working on muscular dystrophy, different aspects, different subtypes, but that's what I've been doing, and I love it. So I was on a job market, just like many other new PIs, and interviewed all over the place, almost Europe, Canada, US. And then along this experience, I learned that I probably would survive everywhere, but I also want to thrive. And a place where I would thrive the most with the kind of research that I do would be a strong hospital, would have strong clinical counterparts who can help me get perspective on the patient side of my research beyond just looking at the mouse model. And children's excels in all of these aspects. So far, I've been enjoying working with my mentors. So my mentors are Jory Broccoli and also being mentored by George Kittis. So they are very passionate in bringing their lab research to clinical application. I also start to collaborate with other scientists here, although we are still ironing out the plans for grads, etc. Like John Alcorn, for example, to evaluate lung functions in the mouse. Also with Rob Nichols on Prader-Willi syndrome, which is also another rare disease. So that sums up all together. Great children's hospital, approachable clinical counterparts, and motivated basic scientists working on bringing therapy for rare diseases. Also, as a kind of like anecdotal story, on the way to Children's from the airport on my interview, I saw the Pittsburgh skyline from the tunnel from the airport, and I just fell in love instantly. I tried a little bit harder to get the position after seeing the beautiful city, because I want to live here. If that makes sense at all.
No,
I was here as a young doctor over 25 years ago, and I've been back here now for 5 years. And that's still one of my favorite views of Pittsburgh, when you come out of the Fort Pitt Tunnel on the way into the city from the airport. And day or night, no matter what the weather is, you have this really, beautiful view of the point of downtown.
Yeah, especially when returning from afar, it's always such a welcoming re-entrance into the city. Well, I'm so happy, Dwi, that you fell in love with the city, because it's exciting to have you here and doing this type of research, in hopes, giving hope to kids who don't have hope now. So I'm very appreciative that you were able to join us today on the podcast.
And that's all for us for this episode. You can find other episodes of That's Pediatrics on iTunes, Google Play Music, and YouTube. Be sure to subscribe to keep up with new content. Leave a review and tell us what other topics you'd like our experts to cover. Thanks for listening.